Oh, should I use your real name when I introduce you, or your code name?
Either works, or both.
All right, both it is.
Okay.
All right, I'm gonna wait for the Twitch ad to clear, and then pause the stream since it's just gonna be stuttery for me on my
Yep.
terrible bandwidth.
Mm-hmm.
All right, I'm ready on my end. Michaela, how about you?
I'm fine.
Alrighty then. All right. Hello, everybody. I am Seamus Donahue of Eve University, and I am joined by HPA Illuminator, also known as Michaela Viking, of the Human Protein Atlas, one of the scientists behind Project Discovery. And this class will be an introductory class about Project Discovery. Uh, this format is a little unusual for Eve University classes since there needs to be a live video component. That's why we are on Twitch rather than on the Eve University Public Mumble. So the way this is going to work is that Michaela will start teaching her class. Questions may be asked in both the in-game chat channel lecture.e-uni and in the Twitch chat. But we're going to let the presentation be given first uninterrupted, and once Michaela is finished, then we will get to answering students' questions. Michaela, go right ahead.
Thank you, Seamus. It's very nice to be here. I'm a bit nervous, but also really excited to see some familiar faces, or at least familiar names and avatars. As you just heard, I'm one of the scientists working in the Human Protein Atlas behind the scenes of Project Discovery. This class today will be really introductory, really basic. We're going to go through the tutorial, see what you can click and not click, and then we're going to do some real samples, and I will just talk my way through them and discuss why I'm choosing certain classes and not others. As you heard, there will be a Q&A in the end, so if you have general questions, please save them until then. If you have specific questions of what I'm talking about if I'm talking too fast, let me know, or if you're wondering why I'm choosing cytoplasm, just let me know immediately and I'll try to answer it. I think there's a delay of between 15 to 20 seconds on Twitch at the moment. And so I'll try to click a bit slowly so you have time to type your questions before moving on to the next example. Okay, as some of you may know, you can restart the tutorial by the little question mark in the bottom left corner. And the first thing that comes up is an info box that the image is the one that should be analyzed, and we should look for the green color in the image. We can zoom the image by hovering over it, and you click the image to fix the zoom in place. So you can zoom in the image, left click, and then you will lock the image. The second box coming up is that there are a lot of categories on the right part of the image. And you should select all relevant categories. Remember, the task is to categorize based on information in the green channel. The categories are divided into nuclear staining patterns, cytoplasmic staining patterns, periphery, MISC, and NA. So the first category is nucleus, which is whatever you see that is overlapping with the blue, pretty much. The cytoplasmic patterns are green stainings that are overlapping with the red pattern. And the periphery are staining patterns at the edge or outside of the cell. Then we have what we call cell-to-cell variations. And sometimes we have images where there is no staining or the staining is very unspecific. There are color channel filters where you can toggle the different colors on and off. I would recommend you to use green only to view the protein, and then toggle red on, toggle red off, toggle blue on, toggle blue off. And when you're finished, you click submit to submit your analysis, and you will get a reward which is based on your accuracy. And you will get a reward in both ISK and in AK, analysis credits, which are loyalty points that you can use to buy certain skins, or not skins, but you can buy lab coat and other things. I see that several people have problems with stuttering in the live stream, and I'm not sure what to do about that. So if someone has some good input, please type it in either Twitch or in the lecture.is-uni chat.
Yeah, we tried to test the Twitch stream yesterday. It was stuttering a lot for me as well, but I just simply assumed at the time it was because I'm on a terrible 2.5 megabit per second download connection. But yeah, if you're having trouble, if this video is stuttering a lot for you, go ahead and say something in either lecture or Twitch. All right, I think these issues are just being experienced by people like myself who have terrible download bandwidth. Go ahead, Michaela. Let's not worry about it now.
Okay, cool. So in the upper left corner, you see your rank, which is rank in Project Discovery. And the bar below is your progression towards the next rank. On the right part, you see your accuracy rating, which is a maximum of 99%. And the higher it is, the higher rewards you get, so it's really tightly connected to the amount of ISK you get. And you can press the help icon in the bottom left corner to replay this tutorial at any time. And if you wonder how many AK you have, you can hover over the ranking and see that you have, well, in this case, 150,000 AK. So, the first sample in the tutorial, we toggle on the green channel to see what it looks like. It seems to be overlapping with the blue, but not with the red. So what you can see in red is the staining for the microtubules, which is the cytoskeleton of the cell. It's kind of like a railway which is transporting things around the cell. The blue channel is a marker for the chromatin or the DNA. So all images will contain the same red and blue markers, but the degree will differ from image to image. The green is... never mind. Let's move on. So since the green is overlapping with the blue, we can tell that, okay, it must be a nuclear pattern. So it's... Here is a bit easier, so you only have 4 categories to choose from. You have the nucleus, which is a sort of smooth or sometimes uneven staining, but it should be overlapping with the blue. We have the nucleoplasm, which is very similar to the nucleus, but it should have holes in them for the nucleoli. And for each of these images, you can also see some other extra examples. Then we can also have some nuclear bodies, which are distinct spots in the nucleus. And you can also have nucleoli, fibrillar center, which are small spots that are overlapping with the holes in the blue. So in this case, I would say that the nuclear staining is quite even. So I will go for nucleus, because I think it looks really similar to these images. And if you're correct, it will be labeled with green. And you can also look at this question mark to see a short explanation of why it's correct or why it's not correct. The second image, we can zoom in on a good cell. Now we have the option of choosing nucleus, speckled nucleus, or nucleoli. And the nucleoli would be overlapping with these holes in the nucleus that should also be seen in blue, which was not very good. Here we have some better. Sometimes you have these holes in the green channel, but you don't have them in the blue channel, but it will still be nucleoplasm. In this case, it's not super visible for all cells, but here you have a really nice staining of a speckled pattern. And I see that Jadarian is asking why this is speckles and not nucleoplasm. And I would say that it's... Well, we decided to have them often mutually exclusive. You should choose either nucleus or nucleoplasm or nuclear speckles. And nuclear speckles is sort of a subcategory of nucleus. It's more distinct. Sometimes there'll be a slight background staining of the nucleus. But if we see a nuclear speckle, we'd rather not annotate nucleoplasm or nucleus as well, just to be as specific as possible. Looking at green channel only, it seems to be overlapping with the blue somewhat. But if you zoom in on a cell, you will see that it is actually also overlapping with the red here. And what you can see is that the red threads are sort of coming together in a centra, and this is known as the microtubule organizing center. So you can see it's sort of like a cluster of green spots sticking together where the red are joined. Microtubule organizing center is very similar to centrosomes. But when we're looking at centrosomes, we really want to see two small dots that are lying next to each other. The microtubule organizing center is usually a bit bigger. And then we also have the ER to choose from, which this is absolutely not, as you can see. The ER is more of like a spider web pattern that is stretching out for the nucleus. So we'll go for microtubule organizing center, or MTOC. And then I was wondering, HBA screener, my colleague, could you answer Albert Whitman in the lecture chat about why it was speckles? Because he would have chosen nucleoplasm too, and he would like a written explanation. Thank you. And here we have a green staining, which is not overlapping with the blue, so it's not a nuclear staining. It's overlapping with the red, which is good. So it's a cytoplasmic staining. We can see that it's not outside the red, it's really contained within the cell. So we still have the ER to choose from, which is looking a bit different. We have the cytokinetic bridge, which is the small junction that ties two cells that have just divided together. And we also have cytoplasm, your favorite category. Once again, we toggle on the green. We can see that it is not overlapping with the blue at all, but it is overlapping with the red. If we zoom in, we see very typical strands that sort of look like spaghetti. It is not cytokinetic bridges. It is not microtubule ends, which it might look a bit similar to. But in this case, it is a very typical mitochondria. And Jadarian is asking why this is not vesicles. It looks like thousands of vesicles. And we haven't gotten to that category yet. Okay, that was for the cytoplasmic pattern in the last image. So hopefully we'll get some more cytoplasmic images or vesicle images so we can look at them. But for the cytoplasm sample, I would say that there were too many and they were too small to be vesicles. So I just want to check that I didn't miss anything, because there can be multiple stainings. So let's choose mitochondria. Here we have a staining that is not overlapping with the blue at all. It is partly overlapping with the red, but we can also see a lot of staining outside the red, which means it's one of the 3 categories in the periphery, where we have focal adhesions. Focal adhesions are sort of like the feet of the cell. They attach the cell to the surface and they help it move around. But focal adhesions should be really short, a bit thick, sometimes almost triangular in their shape. And they're usually located much more under the cell. Then we also have cell junctions, which are stainings between the cells. So that's sort of connecting the cells, enabling them to communicate. But that would only, or should only be seen at the junctions between 2 neighboring cells. So what we're looking at here is not cell junctions, because you can see that it's staining all around the cells. Up here, where there is no neighbor, we still have green staining. So it is plasma membrane. And here we see an example of multiple stainings, which can happen. So we have something that is located in the nucleus, and we have something that is located in the red. So that was not a good cell to look at. I think this is a really good example of when we don't see the holes for nucleoli in blue. You can kind of see that it might be something here, but not really. But the shape of these the structure here is clearly similar to the nucleoli. So even though we don't see the holes in the blue, we can still tell it is nucleoli, because it's really too big to be nuclear bodies. They should be a lot smaller. And then for the green staining, if we zoom in here, you can see the sort of thread-like pattern, which says that it's mitochondria. So we'll click those 2. And the third sample is a nuclear sample. We can see that it overlaps perfectly with the blue. We can also see that if we zoom in, there are holes in the blue, or holes in the green, which are somewhat overlapping with the holes in the blue, but the blue staining is a lot more even. Even so, because we see these very typical holes, we can say that it's not nucleus, it's actually nucleoplasm. And we can also see that there are some variations between cells. So here we have a staining that is much stronger than this one. So we will call it cell-to-cell variations. Here all the examples are from nuclear stainings, but as you might know, cell-to-cell variations can occur for all types of locations. And some locations are easier to tell, some are more different. Nuclear bodies, for example, can be really difficult to tell. Is it really cell-to-cell variation, or is it just that the focus is slightly off? Because they're so small, so sometimes you can't really tell whether they're actually nuclear bodies or not. So here we're getting another tricky one. There seems to be a slight staining in the nucleus, but it's really weak. It's also a very weak background staining in the cytoplasm. And then we also have this very specific pattern next to the nucleus. And we also have a sort of dot-like pattern further out in the cell. In this case, I would say that this is not cytoplasm, nor is it nucleus, because the stainings are way too weak and it's way too uniform. Also, for this case, you can choose it. So what we see here is vesicles, which are small bright spots that are uniform in size and shape, can be evenly distributed in the cell or clustered next to the nucleus, which is the blue marker. They shouldn't be seen in the nucleus. And if they seem, you think like, oh, there are spots in the nucleus, you should turn on the red marker as well to see if you can see some red marker overlapping with the green dots. If you do that, you can think that, well, the focus is probably a bit lower or higher, like lower under the nucleus or above the nucleus, which would mean that the vesicles are outside the nucleus. So here we absolutely see vesicles. And we also see the Golgi apparatus, which is really clear here. You have this sort of dense pattern next to the nucleus. It looks a little bit like mitochondria, but it's very often staining only one side of the cell, or sometimes laying over or around the nucleus. Mitochondria are more spread out through the cell. And up here you also have a very nice Golgi apparatus. I'm so happy I'm not looking at the Twitch chat too much because it's making me laugh all the time. Caleb is being super enthusiastic about everything. Here again we see a very typical nuclear pattern. It overlaps perfectly with the nucleus. And you can also see the holes in the nucleus, which indicates that this is nucleoli. And the reason that these holes in blue are not always seen is that we have about 20 different cell types that we're working with, and some cell types have bigger nucleoli than others, and some have more, some have fewer. So we can say that this is nucleoplasm. It's very easy. And then we also have a staining here, which we can see in red is the cytokinetic bridge. Some people have said that it took a while for them to understand that you should only label cytokinetic bridge when you see the staining in green, not in red. So if you only see the red staining looking like this, yes, it's a cytokinetic bridge, but you should only label it when the staining is actually green in the tip. So here we will label cytokinetic bridge. So we're done with the tutorial. You can always go back to it from the question mark in the bottom left corner. I'll go back to it. This is a foreign cell sample, which means it's a previously unclassified sample. Just as before, we can look at it, but we don't have an answer to it most of the time. Sometimes there will be control samples that we are injecting to see that you're trying your best and also to help you out. So what we can do here is toggle the blue on and off. It seems to be overlapping. We have a slight staining of the cytoplasm. I shouldn't be moving that window. Sorry. Okay, so I have a slight staining of the cytoplasm, which I believe is just background staining, so I will not annotate this as cytoplasm. However, I think that we can see holes in the nucleoli that are overlapping really well, both of these. And also here. So I believe that this is nucleoplasm rather than nucleus. And when looking at the results, we can see that 22% thinks that this is nucleoplasm, about 55% thinks that it's nucleus, and then we have some people who says that it's a negative. And on my screen it looks a bit too strong to be negative. And I do not believe it's nuclear bodies, because if we zoom in, it's a very uniform staining. I mean, it's spots all over the nucleus, not just a few distinct spots. Here we have quite nice sample. We can see that it's mostly not overlapping with the nucleus, so we have a cytoplasmic staining. Let's zoom in. It sort of looks a bit like vesicles here. It's sort of dot-like granular pattern. But for vesicles I would prefer to have less background, so I would say this is just cytoplasm. I think the staining we see in the nucleus is just background because it's too weak compared to the cytoplasm. And this is really subjective in some cases. Is it nucleus? Is it not nucleus? Is it cytoplasm? Not cytoplasm? And I guess it's more difficult for the players because you don't know the gain settings of the microscopes, how much we're amplifying the signal or what laser power we're using. But what you can do is compare it to the other stainings of the cell and see, well, how strong is it compared to the other staining? Do I trust this or not? And you can also always ask other people in the project discovery in-game chat, for example, which is an awesome source of information and cool people to hang out with. Here we see that about a third thinks this is nucleus. I can understand why you would think that, but I would disagree. 88% sees that this is cytoplasm, and then we have 11% that says this is plasma membrane, which was the staining that would go a bit outside the cell. So if we zoom in, it does look a bit here that it's going outside, but it's very minor. So I think it's just the staining being a bit smudgy. Also up here it looks as though it's venturing outside of the cell. But if you compare it to this, you can see that here the staining is very even and uniform, it's really flat. So I would say this is just cytoplasm. It's too... if it goes... what do you say? If it's weaker in the edge compared to the middle of the cell, I would say it's less likely that it's plasma membrane. So when the analysis is submitted, you get experience points, which is based on your accuracy, which is 99%. You get ISK, which is also based on your accuracy. And then you also see what level you are, how many experience points you have, and how much you need to get to the next rank. And when you rank up, you will get analysis credits. Oh, this was a nice one. What you can see here is that the cells in the upper right corner are a bit more in focus than the cells in the lower left. If you turn on the red, you can also see that the cells are looking a little bit damaged, because the red marker is not... it should be like long, nice red strands, but here we see they're a bit chopped up, so the cells are not doing too well. We have a very nice staining of the plasma membrane. It's going outside of the cells. We have very nice protrusions. So I'll click plasma membrane. I think that was a really cool one. Then we also see some green spots, which in some cases seem to be overlapping with the blue here. But if we turn off the blue, I think we will see some red staining behind it, which indicates that this is maybe a centrosome. Also here, it's slightly overlapping with the red. And since we only see one spot, I will label this as centrosome, which should be single or double spots overlapping with the center of microtubules. I do not think that this is cytoplasm because you have a rather even staining throughout the cell. For me to want to annotate cytoplasm as well, I would like to see a much stronger staining outside of the nucleus. And then we can also note that this centrosome is not visible in all cells, but the centrosome is a really small structure, so it might just be due to that the focus is a little bit off in those cells, so the centrosome is a bit higher up. So I will not annotate cell-to-cell variations for this one. So everyone got the plasma membrane. 25% thought this was centrosomes. I think this is looking really good. And we get some experience points, ISK, and the rank is... Well, I'm moving towards a new rank. This was not the prettiest of samples. We see some staining that is overlapping with the blue. And we see some staining that seems to be outside the cell here. This looks a bit like plasma membrane. I'm somewhat hesitant to calling this plasma membrane just based on one cell, but I think maybe this one is membrane 2. And the plasma membrane is the cell membrane which encloses the cell. So in order for things to go into the cell, it has to get through the membrane in some way. If you have small molecules like ions, it can just diffuse through. But if you have larger molecules of proteins, they have to be actively transported through. So yes, I think I will go for plasma membrane here. I will not go for cell-to-cell variation of the plasma membrane because I think that there might be plasma membrane for all cells, just that if we image the cell at the wrong focal plane, we won't see the plasma membrane because it has to be really close to the glass on which the cells are growing for us to see it. Okay, so that's the plasma membrane. I don't think it's cytoplasm. I think the staining we see here is just plasma membrane. We have some nuclear staining though, and the nucleoli are really obvious. They're overlapping perfectly with the hole in the blue. So let's click nucleoli. Then the question is there something else? In this cell, it looks a little bit like there's a nuclear staining too, as well as here. And this one really looks like nucleus. And I think this is kind of difficult because when we image, this image is probably taken to show maybe both the nucleus or the nuclear stainings, as well as the plasma membrane, which means that neither of them is in perfect focus. So I don't know if I really believe that this is a nuclear staining or if it's just like a background staining. I think the nucleoli are really obvious and the plasma membrane is really obvious though, so I'll go for those. I do not think that this is vesicles. You see some spots here, but they're really all over the place. They don't really look distinct. It's more like a background staining. Okay, so pretty much no one thinks that this is plasma membrane. 66% thinks that this is cytoplasm, which I can understand. 11% sees microtubule ends, which is interesting. Microtubule ends are staining in the outer part of the cytoskeleton in red. So in the best of worlds, in the best of stainings and samples, it would overlap really nicely with the red, which I don't really believe it does here. I think this is more of a plasma membrane staining rather than the microtubule edges. And then we have 55% for the vesicles, which I also don't believe in. And then 44% nucleoli. That's pretty good. Okay, so this is a really old sample. I can tell by just saying that these cells are really out of focus. So they were probably acquired back in 2010 or something with our old microscope. You can see that the red is really looking bad. And this is... We have two nice cells here in the middle. Here we have a really nice nucleoplasm staining. We don't see the holes in the blue, but you see them in green. Here, here, and here. And also here I think we see some nucleoli. So it's nucleoplasm, and we also have some cytoplasm outside overlapping with the red here. It's a really ugly staining. It sort of looks like plasma membrane because it's a really smooth edge of the green, but that is just due to this being a really shitty sample, to be honest. And had it not been for the two cells in the middle, I would always have said that this is unspecific. If you look at the two cells in the bottom and the upper left corner, I would say this is really similar to this. You just have this even staining over the whole cell. Nothing is really distinguishable. But here, nucleoplasm and cytoplasm, if I want to be nice. And we can see the 50% said cytoplasm. Then we have a mixture. We have ER, which it is not. It is way too even for it to be ER. It is not cytoskeleton because it doesn't overlap with the red. Intermediate filaments. It's not intermediate filaments either, unfortunately. When I started in the group, we said that intermediate filaments is the sort of category you choose when you say, okay, so it's not microtubules, it's not Golgi apparatus, it's not mitochondria. Okay, it must be intermediate filaments. But it's not intermediate filaments in this case. It's just boring cytoplasm. So here we have a staining that is not overlapping with the nucleus, in blue. It is slightly overlapping with the red, so it could be cytoplasm, it could be plasma membrane. And then I'm curious about these green spots here. They could be centrosomes or microtubule organizing centers. So we'll have a look at that. I also see a question about the abnormal sample checkbox, so I'll answer that before. The abnormal sample checkbox is if you see a staining pattern that is you don't really think fits into one of these categories. You think this is something really new and novel. Let's click it. It's also for if you see samples that are looking really weird in some way. Maybe the red channel is broken halfway through or something. They can also click this. We will have a look at all the samples that are marked abnormal to see if they look okay and what's up with them. I don't know why it's not explained in the tutorial. I think it was a quite late add-on for us. And the reason for that we did add it at all was that one of our categories, rods and rings, which should be here. Rods and rings is a very new category for us. We started with it like half a year ago. Throughout the last 5 years or so, we've collected samples that we called sprinkles that had this sprinkle pattern. And then half a year to a year ago, my colleague found out that, okay, but this is actually a known protein category. They have several different names. One of them is rods and rings. And now we started to annotate them. And as Yadarian points out, nucleoli rim is also a new structure. Nucleoli rim we find up here. It's at the edge of the nucleoli. And this one we introduced even more recently than rods and rings, I believe. And we have even fewer examples of it than we have of rods and rings. So, to go back to the image, the green staining here does seem to be overlapping with all the red threads coming together. It's kind of too big to be centrosomes. I don't see one or two distinct spots. It's more like a random granular pattern. Also here it could be 2 spots, but I think it's a bit too big. But it's overlapping perfectly. So let's call this mtoc. And I was wondering if this is plasma membrane at the edge of the cell, but it doesn't go outside the cell at all. So the question is this cytoplasm or not? Or when looking at it like this, it really looks like plasma membrane. So we're like, it should be plasma membrane. But I really want to call it plasma membrane, but I really can't when it's not outside. So the question is, should I call it cytoplasm or not? And I guess I will not. I will only annotate the microtubule organizing center, because I think that is really specific. I will not annotate cell-to-cell variations, even though we don't see it in all cells, mainly because I don't think we can see the red part coming together in any of the other cells. So I think the reason for us not seeing it in green is that it's really out of focus. And I also see that I'm running out of time. I'm talking a lot more than I thought I would, and this is just taking a lot longer than I thought. And I'm so happy I didn't annotate plasma membrane, even though I wanted to, because this was a control sample, which we can see here. It's marked that we think that it's microtubule organizing center. It says classification results rather than consensus. I think I will give this 2 more samples, and then we'll do the Q&A if we have any questions. And the next time I hope that we can go on Mumbles, we can talk live with each other, which would be really fun. And I really wish we would have nicer samples. But this brings me to this little button, load a new sample, which is new, or semi-new. We introduced it when we started over with the new samples, because we ran out of samples because you were too good and too fast. So let's load a new sample.
Just a quick question.
Yes.
For any of our viewers who intend to go on the fleet at 1800 in 17 minutes, put an X into either Twitch chat or lecture. If you're leaving the class at 1800, if you're leaving this at 1800 because you're going on the 1800 fleet, put an X in Twitch chat or lecture.
I have a question for you, Seamus, then. Do we have to leave the lecture.e-uni chat then at 1800?
I don't believe so. The fleets will, if I remember correctly, fleets don't typically use lecture channel. So if nobody here is going on that fleet, then we can go overtime. That's not a problem.
Okay, cool. So we were lucky. We got a really nice sample. We see something that is overlapping with the blue. I think this was a really nice example. We also see a very nice pattern of cell junctions, which is this staining between the cells. It's sort of like an edge. I believe that it's cell junctions rather than plasma membrane, because it's only visible between the cells and not on the outsides of the cells. So if we look at this big one, we have some staining here, but not the other side, which sort of rules out plasma membrane. So let's click cell junctions, and let's click, well, is this nucleus or is this nucleoplasm? In green, I guess you can tell that this is nucleoli. And also here, you can see this nucleoli here. So I will choose nucleoplasm and cell junctions. And then the question is again, is it cytoplasm or is it not cytoplasm? And I am a bit torn whether it is cytoplasm or not. It could be that it's just background staining, but I will click cytoplasm because I think it's somewhat too strong for me to ignore it. Had it been plasma membrane rather than cell junctions, I would be more doubtful to the cytoplasmic staining, because then I would say, oh, maybe it's just plasma membrane that is sort of leaking through. And it was a control sample, so I was completely off here. Let's see, they agree that it's cell junctions. They don't think it's cytoplasm. And the control sample says that it's nucleus rather than nucleoplasm, which I do disagree with. So I will just start push and take a screenshot of this, so I will discuss it with the others. Okay. Okay, but then we know this should not be counted as cytoplasm. But I still get ISK and I still get AK. And here we do see a really good sample of nucleoplasm. Let's zoom in on this cell. We have the holes in both blue and green. We also have this staining outside the nucleus. It's very typical for the Golgi apparatus. You see how it's located just next to the nucleus on one side mostly. Here it does look like it's located inside it, but it's probably on top or underneath it. So we can see nucleoplasm and Golgi apparatus in all cells, I would say. This is a bit weaker, but it's a bit different focus as well. And the green staining out here is overlapping with the red, so it's just some background thing. I see that Sippy wants to have a disagree on test sample and the possibility to explain why. We don't have that at the moment. What you could do is to report it at Reddit, because we have a thread for that. So if you go to the subreddit called Project Discovery, it is linked from the Twitch account, it's one of the links there. There will be a link to the thread where you can report incorrect samples. And what we want then is a screenshot that includes the ID number, which will be shown in the bottom right corner after you submit it. And we also want the ID number in text so we can copy it easily, and we also want a short explanation of why you think this is incorrect. So let's submit this as nucleoplasm and Golgi apparatus. And I see a question from Albert about what body parts these cells are from, if they're from the skin or from other parts, and I'll just answer it immediately because I think it's a really interesting question. But down here in the bottom right corner you do see the ID that we need to identify this sample. And as I said before, we have about 20 different cell types, and they come from very different organs and tissues in the body. Maybe I should just come back to that later in the question part and discuss about it a bit more. So let's keep doing the samples for 10 more minutes, until 6 p.m. sharp, and then we can do the Q&A. So it's perfectly overlapping with the blue, which means it's a nuclear pattern. We see that there are really clear holes in them, which are thankfully overlapping with the holes in blue. Let's call this nucleoplasm. There is also some staining outside of nucleus, which is overlapping with the red, but I think that is just background staining and not cytoplasm because it's really way too weak. And we can see that 70% agrees that this is nucleoplasm. Some people do think that this is cytoplasm, not so surprisingly. And then we have some people who says that this is plasma membrane, which I don't think it is, because you can see that the green is not going outside of the red at all. This is a new sample, or a new staining pattern we haven't seen before. What we see is that it's overlapping with the blue, so it's a nuclear pattern. But you can also see that it's not staining the whole nucleus, it's really at the edge of the nucleus, which means that it's the nuclear membrane. And the nuclear membrane is pretty much like the plasma membrane or the cell membrane, except for that it encloses only the nucleus. And we have the nuclear membrane in order to sort of keep the nucleus safe, because the nucleus does contain all the DNA. So the nuclear membrane enables this very selective transport of molecules into the nucleus or out from the nucleus. Then we see down here it sort of looks like a nuclear staining, and next to it it looks like a nuclear membrane. So what I'm thinking now is whether this actually is a nuclear staining, or is this just a nuclear membrane that we're looking at in the wrong focus. Is this on top or below the nucleus? Sort of the nuclear membrane bleeding through. And in this case I think it was really difficult to tell. I am thinking about it. The red here indicates that we are below or on top of the nucleus, because you see red threads overlapping with the blue a lot. So I would say this cell is imaged on top of the cell. So for this cell, I don't believe that it's nucleus, I believe that it's nucleoplasm, or sorry, nuclear membrane. For this cell, however, I think we also see a little bit too much red here overlapping with the nucleus for me to believe that it's a nuclear staining and not nucleoplasm. So I will only go for nucleoplasm as for the nucleus, but then we also have some staining outside of the nucleus. This dot-like pattern, which really looks like vesicles. So I will go for this vesicle pattern. It's a lot fewer vesicles, more like this image maybe. So we can see that everyone nailed the nuclear membrane, most nailed the vesicles, and 25% thinks that it's a cytoplasm, which I really disagree with. This is way too weak, and we have so many other interesting staining patterns to click. 50% thinks this is nucleus, which is great. I was really torn between is it nucleus or not. Another ugly cell. So I think this is kind of ugly based on this pattern, which looks like something weird happened there. So we have a cytoplasmic staining pattern. It's overlapping with the red. It's not really going outside of the nucleus, or outside of the cytoplasm. Red. It's going a bit outside here, but I think that's just due to focus or like random staining. And to answer Albert's question of why I didn't go to cell-to-cell variation with the former sample of the nuclear membrane, I would say that I didn't do that because the cells in the upper part of the image were a lot stronger for the nuclear membrane compared to the lower part of the image. So I think that when we imaged it, the focus wasn't perfectly aligned all over the focus, or all over the part of the well that we did image. And I think that the variation that we
Y-
saw. Oh, sorry.
you mean that your instruments are not perfectly aligned all the time? That's impossible.
I guess they are perfectly aligned, but maybe we are not doing it perfectly when we are actually imaging them. And we're talking about micrometers differences that makes a huge impact.
Yeah, I am cracking jokes just to be clear.
Absolutely. I got that part. Thank you for clarifying it, though. Um, oh yeah, the cell variation. I don't think that the differences between the cells were due to cell variation. I think it was just due to different focuses in different cells. There might have been differences with the vesicles that were due to cell variation, and I should have looked better at that. But the nuclear membrane, I don't think was variations. And then I see Blair is saying that we're essentially looking at ultra-magnified GIS, and I have no idea what GIS means. Okay, so we have a cytoplasmic staining here. Then we also have some sort of nuclear staining here, which I'm curious whether it's nucleoli, but it does look like it. It does overlap really well with the holes. And as to comment on Blair's question with the GIS, I think it would be really interesting to see it. I think actually when you look at the protein expression over different tissues, it was shown that brain and testis are the two tissues that have the most diverse expression of proteins. They express the highest number. So it's really cool to look at other body fluids to see the protein expression there. So let's go with cytoplasm and nucleoli for this sample to get back to the image. Okay, so most people agree that it's cytoplasm. Some think that this is cytoskeleton intermediate filaments. I can absolutely see why you would think that, but I think that this staining here is a bit too uniform for it to be intermediate filaments. Intermediate filaments are usually more like tangles, or like if you take a cotton pad and tear it apart, it sort of looks that way. I guess it could be vesicles. It does have like a clustering. Here it could be like a lot of vesicles clustering. Maybe I should have gone for cell-to-cell variations because you don't see any nucleoli in this cell. So I was a bit too fast there. And I don't think that this is a nuclear staining because it's a lot weaker than the nucleoli. So I think it's just a background staining there. So here we have a nucleoplasm. I guess you recognize the holes in the nucleus by now. And you also see a cytoplasmic staining outside of the nucleus. So what I'm thinking now is that maybe it is unspecific, or maybe it is cytoplasm and nucleoplasm. And I think it's a bit too clear. I think I can still distinguish the nucleoplasm compared to the cytoplasm. So I will say that it's cytoplasm and nucleoplasm, but this is really borderline. It's borderline to being unspecific, and it's borderline to not being a nucleoplasm staining, maybe just background. So I'm a bit hesitant. So 80% thinks that this is nucleus, and they're totally missing the holes in the nucleus. Some say that this is vesicles, which I guess could be these big blobs, which are way too big to be vesicles. I think it's just random artifacts from the staining. And actually only 60% thinks that this is cytoplasm, which is a bit surprising. Okay, it's 6 p.m., so let's do one last sample. And it's a real nice one. we have a very nice nucleoplasm. You see the holes in the green. Then in the lower part of the image, we see this spot outside the nucleus. Which is overlapping very nicely with the red coming together, so it could be a centrosome, but I would like to see it in one more cell at least, because we kind of say like one cell is no cell. Up here it looks a bit like nuclear membrane. You see this line here, but again, if it's only one cell, I don't want to annotate it. Then up here though, we do see this outline of the nucleus again. So I guess I will go for nuclear membrane. I should also say that once you click nucleoplasm, you cannot click nucleus because they're mutually exclusive. Also, if you click nucleoplasm, you cannot choose fibrillar center, which are these spots in the nucleoli. And you cannot choose nucleoli rim, which was this edge of the nucleoli. And you can also not choose nucleoli, because we think that they are mutually exclusive. So if you see like, oh, wait a minute, but it is nucleoplasm and nucleoli, well, then you should go for nucleus and nucleoli instead. So let's go for nuclear membrane, nucleoplasm, and let's see if we can find one more spot for it to be a centrosome as well. And here we have the cytokinetic bridge, which is not being stained, so I will not label it. That was the centrosome cell. Maybe this could be a centrosome. It's kind of borderline, but I think it is distinct enough, so I will go for centrosome. Yet again, I don't think I will go for cell cycle, because here you don't really see the center of the microtubule, so I don't know if there's a centrosome or not there. And with that, I think I'm finished with this part for tonight. This was much more fun than I thought it would be. And if you're ready, Seamus, I think we can go over some questions, if we have any.
I think, yeah, let me review the notes that I took.
Yeah.
But I think we were answering questions as we were going along, especially questions that happened to pertain to particular images.
Mm-hmm.
Now, I think you might have already answered this one, but is the nucleoli rim also a new structure categorization?
Oh yeah, that was the Darian's question, and I did answer that. Nucleoli rim is a very recent add-on to our annotations, or our labels.
Gotcha. And this other question, I think we only had a partial answer to. You said that these cells are taken from many different parts of the body, is that correct?
Yes. We have approximately 20 different cell types, and they come from all kinds of tissue in the body. We try to cover the whole body. So we have cells from the skin, we have cells from the cervix, we have cells from the testes, and we have breast cancer cells. I should mention that most of our cells are from cancer. We have one cell line that is a primary cell type, which means that it was taken from a donor and then transformed. And you have to do this transformation of the cells or use cancer cells, because if you just take a cell sample from a person, the cells will only divide for a certain number of times and then die. Also, if you use primary cells or sort of fresh samples, the cells are contact inhibited. So if they grow too close together to each other, they will just stop growing and not divide anymore. Cancer cells do not have this limitation, so they can just grow really closely to each other and then start growing on top of each other. They can divide any number of times. They do get a bit more weird after a certain number of what we call passage doublings, I think you call. Or, well, when they have divided a certain number of times, they will get a bit more odd. They are more likely to have mutations introduced into their DNA, so we can't grow them forever. But you can always freeze a stock of like a low passage number of cells, and then just start a new vial whenever your cells are getting old. I hope that answers the question. Yeah.
So basically, cancer, terrible for people, great for microscopes.
Yes. Yeah. I had another question, comment for that. for us it's very useful to work with cancer cells, but they don't always they don't match perfectly with the tissue in the body. So if you have a kidney and a cancer cell from kidney, they won't be the cell won't be sort of like a perfect replacement. It's a good start, but if you really want to study the real tissue, you should use real tissue samples.
But of course, that's much harder to do.
Absolutely, and you have
So so you take these cancerous cell samples, which are easier to work with, and you hope that they're at least somewhat representative of what normally goes on in a human body.
I would say for most cases they are quite good at representing the normal body, but you have to take into account that it is cancer cells.
Gotcha. All right, now we have an excellent question from Jerry49. Can you please briefly explain the painting and microscopy techniques you use to make these changes? And before you actually get into that question, let me forewarn you that a lot of our students here, myself included, are not cellular biologists, so you might need to use smaller words than usual.
Absolutely, I'll try my best. So we have about 20,000 genes. We know that for sure, pretty much 19 to 20,000. What we don't know is how many proteins these genes code for. And just normally you would say like one gene is one protein. So if you take an average cell, it can have anywhere between 10,000 to 20,000 proteins. But what we are doing is just looking at one protein at a time. The other 19,999 proteins will be there in the cell, but we just don't visualize them. So what we do is we take our cancer cells, we grow them on a small glass slide or in a 96-well plate, if we want to go high throughput. Then we add chemicals that will bind to our protein of interest, and we will let it bind to the protein, and that will wash away everything that's not binding. The cells will have been killed, we will fix them and permeabilize them. And then we will put this little slide with the cells in the microscope and use lasers of different wavelengths to illuminate the different chemicals that are bound to different proteins in the cell. I hope that answered the question. Otherwise, please come back.
All right. I got distracted partway through that, trying to catch up on other questions here. Caleb Irania asks, have there been any tests on other species cancer antibodies?
I'm not sure I'm following the question, so let's...
Well, these are all human cells that we've been looking at.
No, it's not. We have one cell type that is coming from mouse, and then we have also tried cell types from rat and from monkey. But on the HPA Atlas, we have published data for mouse and for mouse. However, the antibodies that we're using are not human antibodies. And we do see a very good correlation most often between proteins in mouse and in human cell types. Sometimes it's not, and I'm not sure if this correlates to how similar the proteins are in mouse or in human, or if it depends on how specific the antibody is for a certain protein.
All right. So one of our viewers is Jay Severas, who comments that he or she does immunofluoresciences on a daily basis. Mm-hmm. And he or she wants to know, do you use hybridomas? I have no idea what those are.
No, we're not. We're using polyclonal antibodies, not monoclonals.
Okay. And Jay Severas also may I ask about the fluorophores? Is that the glowy stuff that you're attaching to these cells to image them?
Yes. To go more into more specific, we use primary antibodies to target the proteins. Then we have secondary antibodies that target the first antibodies. And these secondary antibodies have small fluorophores attached to them. And it's the fluorophores that we can use the lasers to lighten up in order to acquire the images.
Gotcha. So if I understood like I said, I got distracted earlier because I'm also trying to manage questions from Twitch chat and lecture, so I probably missed part of this explanation. But basically, these primary and secondary antibodies, the cells are being exposed to this antibodies. You're washing the cells in these antibodies,
Hmm.
and the antibodies are just randomly working their way towards. Oh, I bumped into something I want to grab onto. Latch.
The antibodies
So
should be specific for their target protein. Sometimes you have some off-target binding when they're binding something else. It does happen.
gotcha. But I'm guessing that also means that some of these secondary antibodies that contain the fluorophores, the glowy stuff, haven't reached a target and they're just floating around in random places in the cell when you image them. Is that also correct?
No, I would say the secondary, as long as the primary antibodies are binding something in the cell, the secondary antibody will always bind the primary one, because that's sort of like a jigsaw puzzle. Those two are always binding each other. The question is more of the primary antibody finding its actual target in the cell.
So you might have a secondary antibody that latched onto a primary antibody that that in turn did not latch onto a target.
Yes.
Gotcha. So we still might have fluorophores randomly wandering around in the cell without a proper target.
Rather attached to a protein in the cell, which is incorrect.
Okay, gotcha. Jay Severas comments, I am actually a biologist, would like to know which ones do you use? Oh, you mean the fluorophores, Severas? Can you clarify that question, Severas?
I see that my colleague answered it in the Twitch chat. He says that we're using Alexa fluorophores, which is the secondary antibodies, and for the primary antibodies, for the markers, we are using Abcam antibodies. And the protein of interest in green is always an in-house produced antibody from HPA in 95% of the cases.
Gotcha. Okay. All right. There have been a lot of questions asked in lecture and Twitch. We've tried our best to try and catch as many of these as possible. If you think your question has been missed, please ask again. All right. I'm not seeing anybody trying to. Yeah, I'm not seeing anybody trying to repeat their questions. All right. So the class proper is now over. As has been previously announced, this is a prototype class. This is our first time trying to teach a class, teach this particular class. So we are seeking feedback on how this class was taught. If you have any if you have any suggestions, comments, constructive criticism about improving the way this class is taught for future sessions, please go ahead and put that into lecture or Twitch chat.
I would like to add also that we would love to do follow-up classes that are a bit more advanced and go through more difficult examples. So if you have samples that you're like, how did you think here, or what is this, or how should I look at this, please take screenshots of them, both with green only, red and green, red, green, blue, blue, green. Include the ID if possible and email them to me, and then we can do a follow-up class looking at those questions only and talk about why something is intermediate filament or not, for example.
All right, I did miss a question from lecture earlier. Schauscher Flower Child asks, in terms of value for your work, not our accuracy level, but the value to the Human Protein Atlas, would you rather have us select a category too much because we feel like it could apply, or do you just want us to select a category if we feel really confident is correct? And to simplify the question, is it if we're going to make mistakes, Is it better to overguess categories or to underguess them?
That is a really tricky question. It's also a really good question. I would say that what we've seen when we looked at your answers compared to our pre-classified answers, that you annotate too little. So I would encourage you to annotate everything that you see that you do believe in. And also, I think we have published an article saying that approximately 60% of all proteins do show double or triple or multiple locations, which means that about half of all your samples are likely only one location, but half of them are multiple locations. So yes,
All right.
I think you should, yeah, I encourage you to annotate what you believe in and preferably several locations.
All right, so better to overguess than underguess. Got it.
Yes.
And again, I will repeat my earlier request. If you think we missed your question, please ask again, either in Twitch chat or in lecture. Similarly, if you think we missed your feedback, please also repeat it in Twitch chat or lecture. We also have a class announcement thread on the Eve University forum. Let me actually go link that up real quick, because we will also take feedback on how this class is taught in that given forum thread. I'm pulling it up right now. Here's the link. Link in lecture. And link in Twitch. So if you have any feedback on how this class was taught, and any suggestions or comments on how to improve it in the future, maybe how to adjust Twitch TV settings, or maybe this would be better done as a slideshow, or whatever other ideas that you have, or I'm hard of hearing, can we get subtitles or something? Any sort of feedback about how to improve the class, please go ahead and put it into that forum thread. Michaela, is there anything you want to add?
I don't think so. In case you haven't seen it, there are some links in the Twitch to different pages I think are good. The subreddit for Project Discovery, the in-game chat channel, the HPA blog. We also have a dictionary, in case you haven't seen it, which contains examples where you can toggle the colors on and off. It's not overlapping perfectly with the Project Discovery interface, because you have a lot more categories than we have in there, but it can be a good start when you're starting out with Project Discovery, just to see what things may look like in more detail.
All right. I'm not seeing any last-minute questions, so I think we're going to dismiss the class at this point. Ladies and gentlemen, Michaela Viking, HPA Illuminator, Human Protein Atlas.
Thank you for listening.
All right, I think we can end the stream at this point.
I will do it in a second.